Hello viewer, this is your first agricultural TV station in Nigeria. My name is Joshua Ishaya, welcoming you to yet another interesting episode of the program Aqua Farming. On the program Aqua Farming today, we'll be looking at the control of fish disease by selection. Of course, you already know that this is the program that makes sure we bring to you everything that is in the water and try as much as possible to analyze them. Stay tuned and take a look.
Selection of fish for resistance to disease may prove, and in some cases has already proved to be an important means of control of fish diseases. Selection work often necessitates transportation of fish from one area to another, and this often leads to introduction of diseases and parasites into the ponds, establishments, and commercial water where previously they were absent. Selective breeding of animals and plants for higher resistance to disease assumes ever greater importance. Often resistant to a specific disease is dependent on a relatively simple mutation involving one or few genes. Any gene mutation is accompanied by change in the synthesis of proteins and may cause protein incompatibility of parasite and hosts. Selective breeding for higher resistance to disease and the data available are therefore limited. Insufficient study of fish diseases, especially the infectious ones, and contradictory observation on immunological reaction in fish have also contributed to these. Until recent, some authors consider that fish were not capable of producing antibodies. The studies of recent years shows that fish do produce antibodies, but not so intensively as the higher vertebrates. Immunobiological reactions of fish are undoubtedly affected to a great extent by water temperature. The higher the water temperature, the more pronounced the reaction. At every low water temperature, the processes of antibody formation are almost impossible. Bacterial activity of organs and tissue have been discovered in fish as well. It is probably dependent on the presence of such antimicrobial proteins as complements, isomes, and propardients. At temperature lower than 10 degrees, the mechanism of excretion and bacterial action of organs and tissues predominate over other immune mechanisms. A mechanism of antibody formation begins to function with increase in temperature. Recent data on fish immunity provides selection breeders with a theoretical background for the breeding of disease-resistant fish strains. Now, let's look at selective breeding of disease-resistant in fish. When a disease occurs in a stock of fish, it does not affect all fish to the same extent. Some fish, sometimes large numbers, are not affected. Scapolos cites this example. In spring of 1950, a small pond in Pate's fish farm was stocked with 665 two-year-old carp, which were kept there until early June. In June, an outbreak of infectious dropsy occurred when the pond was fished out. It was found out that 4% of the fish were either dead or doomed to die. 54% of the fish had external ulcers, typical of the disease, and 42% showed no symptoms of the disease. Even progeny of a single pair of spawns reared under the same environmental conditions did not possess the same resistance. Individual specimens may serve as starting points for selection work. Fish belonging to different strains and forms of any species differ in their resistance to disease. The wild carp, and especially its ammo's subspecies, are known for their higher resistance to infectious dropsy than the cultured carp. The replacement of cultured carp with hybrid form in recent years in a number of Ukrainian fish farms have resulted in considerably decrease in the incidence of dropsy in the region. The same was observed in relation to another widespread infection disease of carp, the air bladder disease. It is widespread not only in the US but in the European countries. Carried out experiments on joint rearing of the cultured carp and hybrid of fourth generation of cultured. The results showed that the incidences of air bladder disease amounted to 78% in the former, while it occurred only about 30% of the latter. Symptoms of illness were more pronounced in the cultured carp than in the hybrid. Considering these examples, we may assume that work on development of disease-resistant strains of fish may have started not only with the selection of specimens possessing higher individual resistance, but also with the development of breed groups of hybrid origin, distinguishable by higher resistance to a particular disease. Naturally, such selection work should be carried out at those fish farms which are always under the effect of the disease. Otherwise, the increased disease resistance of the selected stock will easily be lost. As stated earlier, data are scant on the selection of disease-resistant stocks of fish. Many of these studies undertaken on the problem have either not been carried out through or are still in progress. Only 3 examples of such studies have been carried to conclusion, 2 of which concern infectious dropsy. For a long time, dropsy was considered as a bacterial disease, Aromonas punctata being its pathogenic agent. Subsequently, many scientists have questioned the bacterial nature of the disease and have come to the conclusion that dropsy is caused by virus. Direct evidence of the viral nature of dropsy was obtained several years ago by Yugoslavian scientists. They succeeded in growing the causative agent of dropsy on artificial cultured tissue of carp kidney and infecting healthy fish with it. The second International Symposium on Fish Disease held in Munich in 1965 recognized the virus as the primary pathogenic agent of dropsy, while the punctata and other saprophytic bacteria complicate the cause of the disease. Attempt to create a stock of dropsy-resistant carp were made for the first time by Scapolos. Work on selection of dropsy-resistant carp was stated by him in 1935. Carp not affected by the disease, ponds where dropsy outbreak and occurred were chosen for selective breeding. For 2 successive years, the carp were exposed to large amounts of a punctata with a view to artificially strengthening their natural immunity. Fishes having even the slightest symptoms of disease were eliminated. The offsprings of both selected and non-selected fish were compared for dropsy resistance. The results showed that of the former, only 2% contracted dropsy out of the latter 30% did. No dropsy was recorded among the progeny of the selected spawners. From 1939 to 1943, mortality rates for the ponds, 65 ponds with a total area of 180 hectares, while the progeny from selected spawners was reared, averaged 11.5%, its range varying between 0 and 37%. The average mortality rate for the control ponds, 76 ponds with a total area of 474 hectares, was 57%, range being 4 to 96%. Decrease in mortality was observed in subsequent years as well. From these observations, Kappa-Kloss concluded that resistance to dropsy is inheritable and is possible by means of correct selection to obtain stock possessing relative immunity to this disease. However, it is possible to obtain this effect only where a selected stock is not mixed with any other and where no infection is brought in from outside, either with the water or with the fish delivered to the station. Experience over many years have shown that infection brought into a special selected stock manifests, as a rule, in an acute form, but such complete isolation of the selected stock cannot be maintained in the carp farm situated within the region of the natural habitat of infections uncontrollably brought in either by the wild fish penetrating into the fish farm or by the water supply or perhaps by some other unknown agent. It was suggested by scientists in 1967 that such fish farms should select spawners from breed groups that are most resistant or dropsy and increase their resistance further by means of controlled selection. Such fish farms should select spawners from breed groups that are most resistant to dropsy and increase their resistance further by means of controlled selection. Such work has been started by Kiparnikov at the fish farm of the Northern Caucasus The aim of the first phase of this work was to compare the resistance to dropsy of different breed groups of carp. The following groups of carp have been tested singly as well as mixed groups. The trials for resistance to dropsy were carried out at one of the fish farms. The larvae were reared separately. Fish of different breed groups were combined and reared for 3 years, 1963 to 1965. The fish were marked by fin clipping. However, under the conditions of a hot summer, the fins grew, causing errors in differentiation of breed groups. The results of the test showed that the virus group showed marked differences in resistance to dropsy. The Rosa, Cap, local Scaly, Cap and Don River, white cap proved to be the most resistant to this disease. The enhanced resistance of the above group is apparently connected to their wild cap origin. Besides high resistance to dropsy, the Rosa cap was characterized by generally higher viability, and this cap was therefore selected for breeding at the fish farm of the Northern Caucasus, where outbreak of dropsy often occur in the spring of 1965. 3 years old of the groups had attained sexual maturity and served as broodstock of the second generation, which also was subjected to intensive selection for resistance to dropsy. Though this work is far from completion, it promises great economic possibilities, especially in the Northern Caucasus, where dropsy is a major hazard in fish culture. Now, let's look at breeding of disease-resistant trials. In the early 50s, work was started on the creation of star stock resistant to two widespread bacterial disease, furunclosis and ulcer disease. The initial phase of the work was to test the susceptibility of different fish population. The diseases for these ponds at one trout farm were stocked with fish brought from different bodies of water. Fish affected by the disease were released into these ponds, and the dead fish were fed to the experimental ones. The disease broke out in experimental ponds, causing mortality. In the first year, the mortality rates of the brown trouts in the experimental ponds varied from 35.7 to 65.8%. That of the brook trout fluctuating between 5 to 3 and 99.6%. Thus, the two species were seen to differ in their resistance to the disease. Populations with high resistance were chosen for further selection work. Results obtained in subsequent experiments are not available in the literature accessible to us. Furthermore, on the selection of trout for resistance, a furunclosis had been reported by Ellinger. Diverse geological strains of the brown trout and the brook trouts were intensively exposed to and less resistant individuals were removed by mass selection. Survivors were selected as brood fish for the next generation, which was similarly exposed to the disease. Hybridization brought through crossing more promising strains in some instances improved resistance. Resistant strains were compared with regular hatchery stock under actual hatchery conditions. Results obtained thus far indicate that resistance for furunclosis is being achieved. The work is in progress. Now, let's look at effects of selection work on the distribution of parasite and disease of fishes. For carrying out selective breeding of fish, it is necessary to obtain fish belonging to different populations, forms, subspecies, or species from different regions for rearing an experimental fish farm. It is apparent that pathogenic agent of different disease might be brought in with these fish. The danger of bringing in infection is negligible when eggs are chosen at the initial material for selection. There is only one instance on record where infection was introduced into a hatchery with eggs. The pathogenic agent of whaling disease, one of the most troublesome trout disease, was introduced with trout eggs. It was supposed that the spores of the organism could stick to the trout eggs and be introduced in this way into new fish farm and bodies of water. However, there is no corroboration of the possibility of such introduction. Efforts in obtaining carp eggs under hatchery conditions with a view to propagate them artificially have shown that healthy protein may be obtained from the eggs of diseased fish. It has been demonstrated that such widespread disease of carp as an infectious dropsy and air bladder disease are not transmitted. Germinatively, however, there exists the possibility of introduction of parasite infesting the eggs into new bodies of water and fish farms. Fortunately, such parasites are few. Only one species of Colententrata infesting eggs has been recorded. The danger of bringing it along with eggs is not excluded, and every precaution should be taken while transporting eggs for fish cultural purposes. The danger of possible introduction of pathogens while transferring fry or older fish is more real. Survey have collected a good deal of data on this. The following one is an example from 1958 to 1962. Large number of fried grass carp caught in the rivers of China were transferred to the USS. In spite of all the precautions taken, more than 20 species of the parasite were introduced with this fish. Many of these parasites cause severe epidemics, not only among pathophagous fish, but among other cultured and commercial fish as well. Thus, the cestode came to be introduced into the fish farm of the European part of the USS and Central Asia when it infected the carp and many other fishes, causing high mortality. It has since penetrated into many bodies of water and has been introduced into Ceylon and into Romania and other countries of Eastern European. There are many examples of accidental introduction of pathogenic agent of fish disease, along with fish brought in for selection work. The selection of the cold-resistant strain of carp is based on the crossing of Amur, wild carp, and cultured. In the course of this work, a large number of parasites of Far Eastern origin came to be introduced into the various fish farms. Back in 1936, the first lot of Amur wild carp was brought in, resulting in the introduction of two monogenic tramadoes with parasite, the gills and the settle, and quaquias, and caused severe disease. And at present, it is difficult to find a fish farm where these do not occur. In 1949, some Amur wild carp were introduced into fish farm in northwestern region of the SS, and some of these fish were infected with parasite protozoan, which was unknown in the carp pond of the SS. Long distance transportation, which lasted about 20 days, promoted the development of further infections, and when transferred to fish farms, the wild carp passed on infection to the local spawners in the spring of 1950. A heavy infection caused high mortality among the spawners at the Velikovsky fish farm. 100% of the spawners were lost, and the mortality in Bralosian farm was not less than 50%. The disease was introduced with the stocking material into the other farms and in northwestern region of the USSR, and by the mid-50s, it had spread to the fish farm of the Ukraine, Central Asia, and Kazakhstan. It was proved experimentally that fish exposed to infection for the first time turned out to be carriers of the largest numbers. The intensity of disease is 20 times less in fish exposed to the infection repeatedly, and further exposure helped to develop immunity to the disease. Towards the end of the 50s, a new lot of the Amur wild carp was introduced into the three fish farms of Latvia for selection work. A year severe outbreak of dropsy occurred. It was also found that the large nematode previously unknown in the USS was brought in with that of fish. In 1966, a new lot for Amur wild carp was transferred to Rafsa, an experimental fish farm of the State Research Institute of Lake and Rivers Fisheries. Every precaution had been taken. In the autumn of 1967, were found in gills of carp in numbers of ponds. The parasite had never been found in Rafosa ponds before. It is on record that fish in a number of fish farms were infected by air bladder disease, introduced there with carp broodstock brought in for selection purposes. Thus, any transfer of fish for the purpose of selection is always accompanied by danger of introduction of pathogens, even though every precaution is taken. The Agricultural Research Council of Nigeria (ARCN) coordinates the activities of research institutes and is responsible for supervision, regulation, coordination of research activities and programs of higher institutions in Nigeria. And Professor Hamidou Shaributu, since assumption as Executive Secretary of ARCN in April 2020, has solidified the transformation of agriculture in Nigeria through strategic and meaningful execution of research fundings for improvement of the agricultural sector.
The National Horticultural Research Institute (NIHORT) is one of the 15 agricultural research institutes in Nigeria under the supervision of the Agricultural Research Council of Nigeria. The institute started in 1975 as the Fruits and Vegetables Research and Demonstration Center with the assistance of the United Nations Development Programme (UNDP). It metamorphosed to National Horticultural Research Institute in 1976. NIHORT headquarters is located on a sprawling 350 hectares of land at Jericho area, Ibadan. To have a national spread and outlook, the institute has 3 outstations and 2 experimental stations. The outstations are Mbato, which sits on a massive 810 hectares of vast land located in the humid zone of southeastern Nigeria, Bagauda in Kano, made up of 110 hectares of land in the semi-arid zone of northwestern Nigeria, and finally a 156 hectare of farmland in Otukpo, Benue State.
National Horticultural Research Institute (NIHORT) has developed, registered and released 3 new varieties of tomato which are tolerant to Fusarium wilt, root knot nematodes. They are high yielding, early maturing with good nutritional values. These newly released tomato varieties are Otitum 1, Otitum 2 and Otitum 3, and they are adapted to different agroecological zones of the country. To minimize post-harvest losses, the Institute has developed value-added products from tomato, such as tomato paste, dried tomato slices, tomato wine, as well as tomato juice. The Institute has also developed tuta trap tray to control tomato leaf miner, also known as tomato Ebola. Additionally, biopesticides have been developed in the Institute, known as NIHORT Liptol, NIHORT Ractane, and also the NIHORT Sticky Traps, which are effective in the management of tomato.
Since then, Professor Shaributu has operated as visionary captain by restoring the council's glory with the first ever agricultural television and radio station in Nigeria, with well-equipped studios parading state-of-the-art equipment.
The Agricultural Research Council of Nigeria also has a brand new electronic library to further promote research and visualizations of deep thoughts on agricultural development in Nigeria.
The museum, located in the Senator Abdullahi Adamu Center, is an agricultural museum saddled with the responsibility of keeping records of agricultural findings, discoveries, researches for continuity and history purposes.
The Agricultural Research Council of Nigeria is wearing a new look under the stewardship of Professor Garba Hamidou Shaributu, who says, "Alone, I am nothing, but together, I am something." ARCN,
promoting agriculture.
Many thanks to know that you are still staying through on the program Aqua Farming on this episode where we shared the control of fish disease by selection. My name remains Joshua Ishaya. Thank you for being a part of it. Bye for now.